Welcome, I'm Nikki Kinloch. I am the CEO of Cure Cancer, and it's a pleasure to have you here today. What is a sunny day in Sydney? I'm not sure what it's like where you are, but today we have the privilege of talking to Dr. Kevin Fenix for our 6th Lunch and Learn session of this year. Just a bit of housekeeping before we kick off. There's a chat box at the bottom there for Zoom. If you have any questions, please drop those into the chat and I will weave them into the conversation. And I'll be talking to Kevin for about 45 minutes until 1:15 today. So any questions, please drop them in and we'll acknowledge them and move forward with them. So Dr. Kevin Fenix is based at the University of Adelaide. And every year, 5,000 Australians die from bowel cancer, which is a common type of gastrointestinal cancer, and also the second leading cause of cancer-related death worldwide and in Australia. While immunotherapy could revolutionise cancer treatment, current options are very limited and have minimal effectiveness on bowel cancer. Dr. Kevin is exploring new unconventional immunotherapies to improve the treatment of metastatic bowel cancer, and his Cure Cancer funded project focuses on CIK, which is cytokine-induced killer cell therapy, an area less studied than other immunotherapies. So, Kevin, welcome to our Lunch and Learn.
Thank you for having me.
So before we kick off and talk about the incredible work you're doing, do you explain a bit about how you got into research in the first place?
Okay. So basically, as a kid growing up, I've always been interested in science, and biology was my favorite subject. So I was always interested in biology. I think, as most people who are interested in biology, people were given advice, you should do medicine, but I was like, I don't even want to do the medical aspect. I wanted to do something more, I wouldn't say more interesting, but to me, it's much more interesting to me personally. It's kind of like trying to understand mechanisms of how things actually work. So that kind of like led me into doing a degree in medical research, and I enjoyed it during my undergraduate degree. So I took the research pathway, did my honors and PhD here in Australia, and now I'm here basically doing cancer research.
Wonderful. And we mentioned a few long words in the introduction there as well. So maybe
Yes.
we start to explain that before we kick off into your research. So the first bit is the improvement of metastatic bowel cancer.
Yes.
Can you explain what that means?
Yeah, so basically, when people talk about in any cancer really, when most cancers, when they become deadly, it's really not when you first detect them, right? So usually when cancer is detected quite early on, it's actually highly treatable. We can either do surgery or even do some chemotherapy, and the cancer is usually going to get wiped out and it'll stop progression from happening. So cancer becomes deadly when it progresses. It develops into more what we call metastatic disease phenotypes. So what that word means essentially is that the cancer started to spread. So metastasis is just spreading. In bowel cancer, it's pretty much the main cause of cancer-related death. And like I said before, that's probably going to be true for almost 90% of different types of cancers as well. So metastasis just really means the spread of cancer. And in bowel cancer patients, when you have metastasis, usually it happens in the liver. So basically, because of how the colon, the GI tract, really the gastrointestinal tract works, the liver is usually the first site for metastasis for these patients. And then their survival rate just goes down from colorectal cancer as like early detection is a survival rate of 90%, goes all the way down to 30%. So you can see how much more of a severe disease metastatic colorectal cancer disease is. Yeah.
Absolutely. And we had some pretty shocking stats there. So every year, 5,000 Australians die from bowel cancer.
Mm.
Um, and we're talking about it's the second leading common
Yes.
cause of death
Yes.
worldwide and in Australia.
Yes.
Could you help explain from your knowledge why that might be the case?
Like I said before, I think the main reason for it is because we aren't detecting it early enough. That's the reality of it with bowel cancer. We have now better screens for bowel cancer, and usually that means that the blood stool is usually the first step, and that's being given to people, I think above 50 before, but now I think there's a push to go younger. And the reason why for that is because for some reason, and it's well known, it's been published now for in not just in Australia, but in across different countries as well, is that people who are getting bowel cancer are getting younger and younger. And we don't know the reason why for that. And the problem with this is because bowel cancer is conventionally thought as an old person's disease. So that's why the screening starts at around 50 to 60, right? But and the problem with that is like if you develop symptoms as a young person between the 30s to the 45s to the, like I said, below 50, your doctor will normally dismiss this as, oh, it's probably something else. It's not cancer, right? And usually when all the symptoms start appearing that actually potentially you might have bowel cancer, it's usually too late. So it's already the bowel cancer has already metastasized. So I think trying to get back to your answer, why it's causing a lot of death, I think that's because of the fact that bowel cancer is going undetected, essentially, and it's being allowed to progress just because of how our health system works at the moment. But I know that there's a big push as well to lower again the screening for for bowel cancer using the stool test to 45 now. So I think there's potentially better ways to solve this as well. There's prevention as key. Yeah.
Yeah, absolutely. Thank
Yeah.
you for explaining that.
Yeah.
So I also mentioned some other words,
Yes.
cytokine-induced killer or CIK cell
Yes.
therapy.
Yeah.
Could you explain as in lay terms as much as
Yes.
possible what does that mean?
Yes. So basically, I'll just call them a six cells, so it's easier, so I don't have to spend so much time saying it out. So sick cells or sick therapy really is an immunotherapy. So what we do here is we collect the blood from the patient at the moment, so we draw patient blood, and then we grow the cells in the laboratory, expand them up, and we culture them in a way, and it's in the name. So basically, we expose them to certain cytokines, right? So cytokines are just essentially proteins, or essentially, I mean, if you can think about it, signals, right? So signals that will tell your immune cells found in your blood to differentiate and expand. So basically, that means to grow, right? And we are specific using cytokines. We are able to grow a specific type of cell type, right? That we call a killer cell. That's why it's called cytokine-induced killer cells. So we grow them in the laboratory, expand them out, and basically once we know that they are effective using some certain quality controls, we can then put them back into the patient. So that's the premise of the therapy. It's actually, if you have heard about CAR T cell therapy, it's not that different. The only difference here is with sick therapy, it's we don't do any genetic manipulations to it, right? So it's basically means it's for laboratories like ourselves where we're not massively funded, but we want to help make an impact to our patients because we are a research laboratory, but we are linked with the hospital here in Adelaide. So potentially we see this as a therapy that we can adopt here and give to our patients, that there's not a lot of barriers to actually overcome to get this from research stage to the clinical stage. Yeah. Yeah.
Fascinating.
Yeah.
So when you say there's no manipulation
Yeah.
of those cells,
Yeah.
then they're just they're exposed, then they're
Mhm.
put back into the person's blood. Is that
Yeah.
correct?
Yes. So when I say there's obviously still some manipulation going on because we're expanding them. What I'm saying is we're not genetically modifying our T cells. So basically, or our cells essentially. So basically, compared to CAR T, where there's a genetic manipulation component for it, because you're changing certain aspects to your cells so that that makes them recognize the cancer cells. Sick therapy is you give them certain signals because we know that there's a certain cell type found in your blood that can be expanded that will recognize cancer cells. So it's a different way of thinking, of doing things, but all towards the same goal, essentially. Yeah.
Wonderful, thank you. So you've done that expansion, you've put that blood back into the patient,
Mhm.
and then what happens?
So the idea here is that the sick cells will be able to recognize your cancer and essentially kill them, so eliminate them. Yeah. So
Wow, that's
yeah.
pretty incredible.
Yeah.
That
Yeah.
really is. So, how are you on this research? How far
Yeah.
have you got on that, and
Yeah.
what have you seen so far?
Yep. So far, most of the work that we've done have been done in the laboratory. So obviously there are barriers in place. I wouldn't say it's a bad barrier. It's like it's a barrier in place to keep the patient safe. That prevents us from jumping straight ahead to the clinic. So we have to do a lot of proofing essentially that our therapy will work for these patients and that the therapy that we're going to give is actually safe. So to that end, in the laboratory, what we have done so far is we've shown that, yes, we can grow the sick cells using our own hands, essentially. So not just relying on published works, we can grow them ourselves using published protocols that have been provided out there in the bigger scientific community. So that's the first thing. The second thing is we've characterized these cells and shown that the cells that we have generated are similar looking to what has been published. And finally, what we have also shown is that we can actually show that our cells can actually kill cancer cells. And how we do this in the laboratory, we actually use a system which I think you might have heard of before because it's kind of like a cutting edge technology, but a lot of people have adopted it, which we've been starting to use patient-derived organoids. So basically what that means is like when a patient that we see here at the Queen Elizabeth Hospital in Adelaide undergoes hopefully curative intent surgery for them, that means that they take out the cancer from the patient. We just get a little bit of the tumor samples. So we get the blood to create the sick cells, but we get a little bit of piece of the tumor as well. And we expand them in the laboratory using this cell culture technique called organoid cell culture. So what that means is that we are actually generating sick cells from the patient and testing them using the same cancer that has been growing inside the patient and showing that there is quite good efficacy going on when we test our cells against these cancer cells in the lab. So that gives us confidence that this therapy may work, and we are hopeful that we can actually jump into clinical trials quite soonish. But like I said before, there are still multiple hurdles that we still need to handle. But at least for us, we can show now that we can generate them, they can kill the cancer cells, which are the two most important things, I think, for us.
Yeah, absolutely. And those organoids that you're growing outside
Yes.
of the body,
Mhm.
essentially you're testing the effectiveness of it without
Yes.
having to test on the patient.
Yes, exactly. So that's the whole point, is like the idea here is that hopefully it's much more clinically relevant than using cell lines that have been generated 50 or 60 years ago that's been expanding in the laboratory. So we don't even know if it actually looks like the cancer that currently patients are having. So this is much more clinically relevant, I guess.
Yeah, absolutely. I'm just going to pause there and welcome had a couple of new guests. I'm Nikki Kinlock. I'm talking to Dr. Kevin Fenix today at the University of Adelaide on his immunotherapy work on bowel cancer. If you do have any questions, drop them into the chat box there at the bottom, and I'll weave those into our conversation. So we've talked about how you're taking out the blood, you're expanding the cells, you've got sick therapy treatment, so you then put the blood back in.
Yep.
Those cells then go and seek the cancer cells
Yes.
and
Yes.
look to kill them.
Yes.
And the results you've seen from taking tumor biopsies and testing this therapy on that tumor cell outside of the body have been pretty positive so far.
Yes. Yes.
Lovely. So a question about who would be able to get this therapy treatment. Is it based on how advanced your cancer is, your age? Are there different brackets that you work towards on this?
So for us, for our team, we're really developing this therapy for patients that have severe prognosis. So basically, like I said before, metastatic patients. And the reason why is because, as I mentioned before, if you get diagnosed with primary bowel cancer, the treatment for it works really well. Survival rates of above 90% now. So in that sense, for us, what we really want to do is provide a therapy for patients who actually need something else, need some hope to actually, say that they can actually beat this cancer. So that's our target cohort.
Yep.
So but in terms of actual sick therapy, we don't see it as something that won't work when you give them at the primary disease. What we're just saying here is like, well, we could give it to those patients, but because there are other treatments already available, then it doesn't make sense for us to push it at that angle. We prefer to push it towards giving it to patients that have metastatic disease, just because of that reasoning. Yes.
Yeah, absolutely, and that makes sense.
Mm.
Would it be fair to presume then that as the altered blood goes around your body,
Yes.
it will be targeting not only the cancer cells that you'd be aware of, but it would be looking for other cancer
Yes.
cells that might not have been
Yes.
diagnosed as of yet as well?
Yes. Yeah. So that's kind of like, I guess the beauty of immunotherapy is like you're kind of like providing a living therapy essentially. And we don't know the answer to that yet because we haven't tested it in humans, or obviously, or so we don't know how long these cells persist in the body. So we know that when we transfer them, hopefully there's going to be some efficacy towards the cancer cells at the primary site, but it is a systemic injection. So basically that means it's we just reinfuse it back to the blood. So there's opportunities there, as you've mentioned, and this is why we said metastatic disease might be ideal for this, is that it can go to multiple sites, no problem, and find those cancer cells that we might not even know of. So I think that is a good point to highlight as well. Yeah.
I mean, that would be incredible, and particularly for people where it has spread so much that
Yes.
the other therapies aren't going to work as
Yeah.
well.
Yeah.
Yeah. What about a question here about comparative treatment? So, I would imagine this kind of treatment would be less invasive for
Mhm.
patients as well, as opposed to chemotherapy or other treatments in regards to physical outcomes. And obviously, I appreciate you haven't tested on humans, but the
Mm.
idea that this was actually going back into your blood would
Mhm.
be less difficult for a patient to deal with than other drugs being implemented into their body.
Mhm. Yeah. I mean, so I think that's that's a good thing that you've touched upon. So just to get to one of the reasons why we were interested in adopting these technologies was like there have been clinical trials happening across the world as well. And people have reported the clinical outcomes from these patients. So obviously, other than improving their prognostic or survival over time compared to not having this therapy, one of the things that came out from these studies as well was that sick therapy doesn't have a lot of side effects. So that's very important for us. And that's quite ideal for us because I'm not sure if you know, but immunotherapy, like the standard ones that we give at the moment, like checkpoint blockade and potentially even CAR T cells, have a lot of severe side effects that they might get overactivated that happens to these patients. So obviously it's a big problem that people are trying to solve. But what we found that was interesting for us was the sick cells um, weren't having as much problems with side effects. Now, does this mean that it's going to be better than what's already out there? Well, we don't know the answer to that. And the second thing is like with how we're designing our experiment so far anyway, we think that as a first step, seek therapy based from our previous studies, we probably want to combine it with chemotherapy still initially. But potentially what could happen is if we do see efficacy by combining these two things together, is we could potentially reduce the dose of chemotherapy. So potentially reducing side effects. But like I said before, that's probably quite a long-term goal. Like for us right now, we just want to see if we give a patient their standard SOP or standard protocol for how they're normally getting treated, usually with chemotherapy. If we just add sick therapy, can we improve clinical outcomes by survival without inducing any more harsh side effects? So yeah, but one of our goals as well for the research that Cure Cancer is funding is to see if we can actually make sex therapy a lot more effective in the way that in the future, potentially we might not even need chemotherapy. We can just give this some monotherapy.
That would be incredible. You touched on how they're trialing this in other countries. Is it the exact the same therapy? And if so, that would give us hope that we could get through the ethics
Yes.
rounds here in Australia much quicker.
Yes.
And get it into patients as
Yeah.
well.
Yeah, so basically that's true. So it's been trialed globally. But the main supporter of this therapy is in China.
Mm-hmm.
So, but it has been put into patients. So it's been put into patients quite a lot. So we recently did like a systematic review. So it's a type of research where we collect different data from multiple published papers and unpublished, but released data sets, clinical data sets, and have shown that at least in the bowel cancer space, about 5,000 to 7,000 patients have actually received this therapy already. And like I said before, we haven't seen any at least not what they call that, adverse events. So basically bad side effects having been reported to these patients. So that gives us confidence that potentially we can jump through the hoops of, like I said, the barrier to the clinic a lot quicker than normal. Yes.
That would be incredible if you could as well. I have a question here that says, how long will that be? But I'm
Yes.
not sure you know the answer, would you?
You know what they say, how long is a piece of string? But with that being said, I mean, I wouldn't say we are uniquely positioned, but we are positioned in a way that our team is led both by myself as a research scientist, but also is being highly supported by the oncology team here at the Queen Elizabeth Hospital. So really for us, the question is like, how long will it take to get to the clinic? We're aiming for within the next 2 to 3 years. That's our goal. So there are certain things that we need to still overcome, so that we're trying to overcome this year. So basically, two important aspects is we've said we can grow the cells now, but we're growing them in small scales fit for the laboratory. We actually want to grow them in larger scales now. So basically, can we actually get patient blood and grow them to or expand them enough so that we can actually give them back to a patient? So that's the first thing. The second thing is, which is critical for ethics, is can we show that our 6 cells that we've generated ourselves is actually going to be safe for the patient? So we're basically doing some testing to confirm that these 6 cells won't induce some kind of severe adverse event, essentially, using different models.
Okay, yeah, that makes sense. Thank you.
Yeah.
A question about blood types. Does this therapy work on all blood types?
That's an interesting question. We haven't done specifically looking at that. All I can say though is we have recently published a paper. Like I said, we published a paper where we've tested out basically generating 6 cells in the laboratory from different patient blood, so both colorectal cancer patient and also healthy donors. To answer your question, 90% of the patients that we have collected from generated 6 cells in high efficiency, so they've expanded quite well. There is that missing 10% that failed to produce 6 cells. We don't know what's causing that. Blood type could potentially be an answer, but we haven't really looked at that when we went there, when we looked previously. Yeah. Yeah, but we do just to touch on that question as well. I was recently in Europe to a laboratory that's looking at sick cells, and that's something that they're exploring. It's like HLA, so blood type. How does it actually impact sick cell production? And they seem to think that there might have it. There may be an effect, but they're not conclusive yet.
Yeah, absolutely. And do you have much to do with the patient from a clinical side? And if so, how does talking to them or meeting with them
Yes.
affect your research?
Yeah. So initially, when I first started my role here at the institute, at the Basel Hetzel Institute, which is the hospital research institute here at the Adelaide, I was much more involved because I was much more junior in my role as a scientist. I just finished my PhD. So that meant that I was more involved in actually consenting patients. So basically, obviously for studies, we can't just take blood and use it. We have to make sure that the patients themselves are happy to contribute their blood to these patients. So back then, I was much more involved with interacting with patients. Now that I'm more senior in the role, I'm mostly in the laboratory side of things. And also, I wouldn't say the regulations have changed, but we have changed our system now in terms of like our clinical leads, because it's their patients, they talk and consent them now. So it's much more easier, because like I feel like before it's a bit, I wouldn't, the patients that I have consented based on previous experience have all been fantastic, and I've always been moved by that fact. But I think there is still, like, I've always felt that, some stranger approaching you to talk about something else, whereas the clinician, their actual doctor, talking to them, it's much more, I guess, like, to me, if I was the patient, I think it would be much more willing to cooperate. And like I said before, 100%, I haven't had anyone, when I was working as consenting these patients, I haven't had anyone say no. So it's not a big issue, but I think it's kind of like a thing that, yeah, that helps. But yes, so I have interacted with patients before, and it has shaped my research as well, because like I said before, I think just like I said before, like, thinking back, if it was me, would I be as generous as these patients are? And I wouldn't know the answer to that question just yet. I think you have to experience it, and that's why I thought it was quite and it's a moving experience being able to interact with patients, and really it's humbling experience as well. And I think and it's really moved me to find new treatments for these patients, because, like, I mean, the patients that I saw were the patients that were essentially getting cured, quote unquote, because, like, it is the surgery is the final curative treatment for liver metastasis. So I guess maybe it's it's biased in that way, but, like, even them, they were all quite generous in supporting research. I'm quite happy with that. Yeah.
Lovely, thank you. A question on whether you take the blood out of a patient and you then do this expansion and you put
Yeah.
it back. Is the ability to be able to to give the blood to someone else? Do you have to do in your hypothesis, do you
Mhm.
back to the same person, or can you
Yeah.
share that blood with
Yeah.
other patients?
So that's a good question again. So in our therapy, yes, you can eventually do that. So eventually you can get blood from healthy donors even. So we don't even have to use the cancer patients to expand the cells and then give it to patients. At the moment, we are just working on what we call autologous, so coming from the same patient, right? But in the future, we could potentially give sick cells from, like I said before, we probably wouldn't give it from one cancer patient to a different cancer patient. But potentially, what we could do is get blood from a healthy donor, expand them, and then give them to a cancer patient. Because we have done some tests, like, it's not the best test yet. So potentially in the future, we might look at this again. But so far from now, we know that getting blood from a healthy patient and a cancer patient, we can grow sick cells in the same rate, and they seem to be quite effective in killing cancer cells. Yes. Yeah.
Lovely, thank you. Okay, I have a question here that's clearly from someone scientific, so excuse me as I work through these very long words. But do chronic imbalances in the intestinal microbiota populations and inflammation of the intestinal epithelial barrier caused by lifestyle and dietary factors
Mhm.
a major risk for the development of bowel cancer?
So trying to digest that and trying to, like, simplify that. is the question here is like, is gut microbiome imbalances affecting bowel cancer initiation?
Yes.
The answer to that probably is yes. There's a lot of research going on now where we know that a lot of cancer patients have dysbiosis in their gut microbiome. I wouldn't be 100% sure that that is actually what's causing the initiation, but we do know that a lot of bowel cancers develop from inflammation. So there might be a link, but I don't really do much research on the initiation of bowel cancer, so I can't give you 100% answer to that question. All I know is like there are links with dysbiosis with inflammation, and inflammation is critical for bowel cancer initiation. So at least certain subtypes of bowel cancer. So there
Lovely.
are links there, but like to definitely prove it, I wouldn't know. You have to ask someone who was actually working on
Yeah.
that type of research.
That's
Yes.
absolutely lovely. Thank you. And another question here about could certain strains of probiotics and colostrum immunoglobulins have a protective effect against bowel cancer?
Probiotic bacteria. I Well, there seems to be. I think these are all at the moment quite speculative. Uh,
Mm.
that if you have probiotic bacteria, um, that's gonna be better for you. But there are a lot of research on it already. So like, people are performing fecal microbiome transplants as a way to enhance treatment outcomes for not just bowel cancer patients, but for a lot of different types of cancers. So I think the answer to that question is it's still up in the air until we actually get the clinical trial results from those type of work. Yeah.
Yeah, lovely. Thank you. Okay, getting back to your day in the life. So what does a typical day for those that
Yeah.
don't normally meet researchers? What
Yeah.
does it look like?
Yeah. So it's actually quite I would say it's different depending on what stage you are. So I'm a bit more senior now. I'm like 6 or 7 years post my PhD. So that means that I'm actually much more of a supervisor than actual someone who works in the laboratory as much. So I still go to the laboratory, but usually before, when I was much more junior, I'll go to the laboratory because I was the one performing the experiments. Now I go to the laboratory because I have to show someone a certain technique, but usually I have junior researchers such as PhD students or even young postdocs, so basically people who just finished their PhD, so we call them postdoc or research assistants who need some training or need some help on how to do a certain technique. But day to day, my main roles are really, it's really just I wouldn't say it's just an office job. It's partly an office job where I'm in the computer. I'm either writing grants, which is consumes a lot of my time. So it's a lot to continue on attracting funding, hopefully to the research so we can continue on year. That's one thing. The other thing that I do is obviously write or edit papers. So basically, we as scientists, but like we must disseminate our research. So basically, that means that we have to send our findings out into journals. So basically, we as a publication. So basically, it's a lot of writing involved in terms of like editing manuscripts, formatting data, making sure that it's, I guess, readable, but also it makes sense and everything else like that. So there's a lot of that. Like I said before, it's quite mixed actually. So there's a lot of like involvement with student supervision as well. Not just my students, but also students at the institute where I'm being asked to like, help out with marking their presentations or their other work. So it's quite varied.
Yeah, lovely. Thank you. We've got another 10 or so minutes with Dr. Kevin Fenix at the University of Adelaide. So if you do have any other questions, please pop them into the chat and I'll include them in our conversation. What would you say your biggest challenges being a research scientist?
I think the biggest challenge for me, and I think it's not just for me, but it's a lot of researchers, is essentially obtaining funding to continue doing research. So I think Cure Cancer is a big supporter of this idea that early career researchers, but any form of researchers really these days struggle to attract funding.
Mm.
And why is funding important? Well, funding it's important. It's because it's the funding that you provide is not just allowing us to actually do experiments, but it's actually paying for the salaries themselves. So without funding, we can't continue, literally. And I'm not sure if you guys are aware, but every year, yes, the government has been quite helpful in providing funding, but the funding hasn't really changed over the last 5 to 10 years.
Mm.
And what that means is that the success rate of winning a grant has gone down from, I don't know, before apparently it's around 20%, but now it's 10%. So that means it's a 1 in 10 chance that you get a grant and that those grants maintain your salary. So that means that government grants are quite hard to get, but then also that means that we have to rely on other people who are actually supporting of research, just Cure Cancer and other charities that actually allow us to continue doing research. Because like I said before, like without funding, which is quite happening to a lot of people as well, is then you can't do research. So people have to decide, okay, what am I going to do now? I have to change careers or change topics or something like that. So I think that is a big challenge that as any researchers who find in Australia at least that the lack of job security is a big problem. It affects the mental health of people essentially. So yeah.
Yeah, of course. And obviously, I mean, this is why Cure Cancer exists. For the last 57 years, our focus has been on funding young researchers. So ideas like yours
Mm.
can get off the ground,
Yeah.
and you can have the funding
Mm.
to look at doing things differently as well,
Yes,
making
exactly.
progress.
Mm.
And you're right. I mean, even in my tenure here at Cure Cancer, which is 6 years, I've seen many researchers leave research.
Mm.
I've seen many cancer researchers go to mental health research because they get a lot more money in mental health
Yeah.
research. So it's one of those really catch-22s that you're on the cusp of scientific greatness and developing this thing, and in fact you can't even guarantee your salary to be able to
Yes.
do that, which is such a shame, isn't it?
Yeah. Mm. So I think advocacy for better I guess advocacy just for awareness of this, and hopefully that means that in the future there might be actually some meaningful change, but
Yeah.
to improve these type of stuff would be fantastic for researchers, not just for myself but for everyone.
Absolutely. Thank you for sharing that. What motivates you then? It's obviously not the continuation of funding, but but what motivates you to carry on doing what you're doing?
Yeah. Well, I guess to me, just in general, I think I like what I actually do, you know. I like the fact that I'm doing something that potentially no one has done before. So
Yes.
I think a lot of scientists will tell you the most important thing to become a good scientist is curiosity. So being passionate about actually investigating something. And I think with all the downsides of this career, there's a lot of upsides as well. So one of the things, like I said before, is like you can potentially answer something that no one has answered before. And for certain type of people, that's actually enough to motivate them to continue on. And it motivates a lot of people. That's why our things are still going on properly. But the other thing that I would say is like, because I'm working on translational research, the fact that potentially my research might actually lead to some kind of impact to certain people, to me, that's also quite motivating as well. Yeah.
Yeah, absolutely. And it's I like that idea about the curiosity, because we talk about that a lot with other researchers, that even if you do a piece of research and it doesn't end up with the outcome you expect,
Yes.
it's still a learning, it's still a
Yeah.
positive as
Yeah.
well. So yeah, absolutely. So a quick question here on metastasis again. So wondering where primaries have been. Does it matter? So from cancer that metastasizes, does it matter where the primary starts from in regards to your treatment?
Um, I guess like for us, um, we look at it from a different perspective. So basically, I'm talking about this from a sick therapy point of view, but then I'll also talk about it from a general metastasis point of view later on. So how our sick cells work, they actually use a different cancer recognition mechanism that actually is generalizable to any type of cancer. So the receptors that we're looking for are found in majority of different types of cancer. So that means that whether it's a metastasis or a primary, personally, I don't think it would matter. Our therapy should, I'm not saying it will, but like it should work. And because of this, sick therapy has actually been trialed for multiple different types of cancers, not just in bowel cancer around the world. So obviously blood cancers is one of the things that people have used this quite often with. So like in lymphoma, for example, but people are also trialing it for breast cancer as well. And for other solid tumors, like I think prostate might be one of the big ones as well. But and obviously bowel cancer is what we're looking at. So from our perspective, using our therapy, I don't think metastasis is the origin of the primary site. It's not going to be as an important factor.
Mhm.
However, if you're talking about actual current ways of how we treat cancer patients with metastasis, then yes, the origin of the primary will be important because it dictates how the patients actually receive different types. People know that even different types of chemotherapy will work a lot better.
Mm.
So there are different types of chemotherapy regimens. So when we say chemotherapy, it's a general word, but there's actually different types as well that are being given to these patients. And there's obviously also targeted therapy now that are being developed that's specific towards a certain cancer. So which basically what I mean by targeted therapy is like they're targeting a certain signaling mechanism that has been overused by the cancer cells. So we're specifically targeting that cancer. And we know that certain cancer types will have different signaling pathways that they are overusing. So different targeted therapies for different cancers. So then what the primary site for the metastasis is going to be important. Yeah.
Yeah, lovely. Thank you. And one final question before we wrap up. What are your hopes for the future in the research that you were doing?
Well, I'm hoping that we can actually get into clinical trials in my lifetime within the 3 years, like I said before, of this research, because I think, like I said before, I'm hoping, and also not only that we can get into clinical trials, but we can actually hopefully see the positive outcomes that people have been reporting across around the world. So like we would like to see that as well replicated here in Australia, hopefully. Yeah, that's my goal anyway. But yeah, but we're doing some other stuff as well that for me it's much more. I would like to continue on doing curiosity-driven research as well, as much as like translation research is important, but I do think that basic fundamental research where we're actually just asking questions because we're curious about it there still should be a space for it and we can still continue doing those type of things because like that's how we're gonna find new therapies is through unexpected findings. Yeah.
Absolutely. Well, Kevin, it's been a pleasure to chat to you today. Thank you for your insights. Thank you to you and your team for the work
Thank
that
you.
you are doing as well.
Yeah, thanks.
Thank you to our audience. I hope you enjoyed that session with Dr. Kevin Fenix at the University of Adelaide. Please join us at the same time on the 15th of August for our Lunch and Learn with Dr. Arutha Kulasingi. He's at the University of Queensland, where we will be talking about his work on lung cancer and immunotherapy. So until then, stay safe and have a good afternoon. Thank you.